Genetics is the study of how living things inherit traits from their parents and how these traits affect their appearance, behavior, and health. Genetics also helps us understand how life evolved and diversified over billions of years. But how do we study genetics? What are the tools and techniques that scientists use to unravel the secrets of life? And what are the ethical and social implications of manipulating the genetic code of living organisms? DNA, or deoxyribonucleic acid, is a molecule that contains the instructions for making every living thing. DNA is made up of 4 types of building blocks called nucleotides: adenine, thymine, cytosine, and guanine. These nucleotides pair up in a specific way, A with T and C with G, forming a double helix structure that looks like a twisted ladder. The order of these nucleotides along the DNA strand determines the genetic code, which is like a language that tells the cell what proteins to make and when to make them. Proteins are the molecules that perform most of the functions in living cells, such as catalyzing chemical reactions, transporting substances, fighting infections, and regulating gene expression. DNA is not just a long string of nucleotides. It is organized into units called chromosomes, which are tightly packed bundles of DNA and proteins. Humans have 23 pairs of chromosomes in each cell, 1 pair inherited from each parent. Each chromosome contains thousands of genes, which are segments of DNA that code for a specific protein or function. Genes can be turned on or off by various factors, such as hormones, nutrients, stress, or other signals. This process is called gene expression, and it determines how cells behave and differentiate into different types of tissues and organs. Gene editing is a technology that can change DNA sequences at 1 or more points in the strand. Scientists can remove or change a single base or insert a new gene altogether. Gene editing can literally rewrite DNA. It is performed using enzymes, particularly nucleases that have been engineered to target a specific DNA sequence, where they introduce cuts into the DNA strands, enabling the removal of existing DNA and the insertion of replacement DNA. One of the most powerful gene editing tools is called CRISPR-Cas9, which was discovered in 2012 by scientists who studied how bacteria defend themselves against viruses. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are sequences of DNA that bacteria use to recognize and destroy viral DNA. Cas9 is an enzyme that acts like scissors to cut the viral DNA at the CRISPR target site. Scientists realized that they could use CRISPR-Cas9 to edit any DNA sequence by designing a guide RNA molecule that matches the desired target site. By attaching this guide RNA to Cas9 and delivering it into a cell, they can direct Cas9 to cut any gene they want. Gene editing has many applications in different fields of science and society. For example, in basic research, it can help scientists study how genes function and interact with each other in different organisms. By creating mutations or inserting new genes into yeast, bacteria, mice, zebrafish, and other model organisms, scientists can discover how genes affect physical traits, development, behavior, disease susceptibility, and evolution. In biotechnology, gene editing can help scientists create new products and processes that benefit humans and the environment. For example, it can be used to engineer bacteria or yeast to produce biofuels, drugs, vaccines, or other useful substances. It can also be used to modify plants or animals to enhance their productivity, quality, or resistance to pests and diseases. In medicine, this can help scientists develop new treatments for genetic diseases or cancers. For example, gene editing can be used to correct faulty genes in cells or tissues that cause diseases such as cystic fibrosis, sickle cell anemia, or hemophilia. It can also be used to modify immune cells to target and kill cancer cells more effectively. In bioengineering, gene editing can help scientists create novel organisms or traits that do not exist in nature. For example, it can be used to create glowing plants or animals, transgenic animals that carry human genes or organs, or designer babies with enhanced or altered characteristics. Gene editing is a powerful technology that has the potential to improve human health and well-being, but it also poses significant challenges and risks. Some of the ethical and social issues that need to be addressed include: gene editing can have unintended consequences or side effects, such as off-target edits, immune reactions, or genetic instability. These effects can harm the edited organism or its offspring, or even affect the ecosystem if the edited organism escapes or reproduces in the wild. Therefore, it needs to be carefully tested and monitored for safety and efficacy before it is applied to humans or the environment. Gene editing is subject to different laws and regulations in different countries and regions. Some countries have strict rules that prohibit or limit gene editing in humans or animals, while others have more lenient or unclear policies. There is a need for international cooperation and coordination to establish common standards and guidelines for gene editing research and applications. Gene editing can be expensive and inaccessible to many people who need it. There is a risk of creating inequalities or disparities between those who can afford or access gene editing and those who cannot. There is also a risk of creating a market for unproven or unsafe gene editing products or services that exploit vulnerable or desperate people. Therefore, gene editing needs to be affordable and accessible to everyone who needs it, and regulated to prevent fraud or abuse. Gene editing can create social or ethical dilemmas about who should benefit from it and who should bear the costs or risks. For example, it can raise questions about human dignity, identity, diversity, and rights. Should people be allowed to edit their own genes or those of their children for cosmetic or non-medical reasons? Should people be allowed to edit their genes to enhance their abilities or traits beyond the normal range? Should people be allowed to edit their genes without their consent or knowledge? How should society treat people who are edited or not edited? How should society balance the interests of individuals and groups in gene editing decisions? These are some of the questions that need to be considered and debated by all stakeholders involved in gene editing. It can generate public fear or mistrust due to lack of awareness, education, or engagement. Many people may not understand how gene editing works, what are its benefits and risks, or what are its ethical and social implications. Some people may have religious, cultural, or personal objections to gene editing based on their values or beliefs. Therefore, gene editing needs to be communicated clearly and transparently to the public, and involve the public in dialogue and decision-making processes about gene editing research and applications. It is a technology that can change DNA sequences at specific locations in living organisms. It has many applications in different fields of science and society, such as basic research, biotechnology, agriculture, medicine, and bioengineering. However, gene editing also poses significant challenges and risks that need to be addressed ethically and socially. Gene editing is not just a scientific issue, it is also a human issue that affects us all. Therefore, we need to learn more about gene editing, engage in constructive dialogue about its implications, and participate in responsible decision-making about its use. Thank you for watching this video. If you liked it, please give it a thumbs up and subscribe to my channel for more videos on science and discovery. And don't forget to leave a comment below with your thoughts on gene editing. Thanks for watching.